Hi and welcome to QSD. You may well have heard of the microbiome in recent years. Indeed, I have already covered it on my young channel. And you may well be starting to appreciate just what a huge part it plays in so many areas of your daily life. And well, it seems like things may well have just entered a whole new level. Researchers have finally managed to start observing and therefore understanding a whole new layer of complexity previously too small to comprehend. So, does that fire your interest? Keep watching if you want to find out the who, the what, the where, and as always, at the end, I'll give you my own personal thoughts on the subject and how it relates to the bigger picture. We all know we share our bodies with literally trillions of bacteria, inside and out, and that our good health, both physical and mental, are intricately intertwined with a delicate balance to it all. Indeed, knock back one variety and another will flourish. Eat a type of food and another will thrive. Indeed, take one from one area and introduce it to another region of the body and all hell can break loose. Literally, well, figuratively. In fact, it could be argued that life is essentially trying to keep the precious microbiome that you were given in time up to and including your birth. And then it kind of morphs into a quest to find and protect the ones that keep your systems in balance and conversely to avoid the ones that will do the opposite. Think about it. Anyway, I digress. Researchers at Stanford University School of Medicine have finally created an approach to read the tens of thousands of proteins so small that they have gone unnoticed in previous studies. These proteins belong to more than 4,000 new biological families, and they will be the instant messaging system of what we have so far just swept under the proverbial carpet. We can be sure that these will include, among other processes, the warfare waged amongst all the different bacterial strains as they vie for survival and seek to replicate in their coveted biological niches, alongside the cell-to-cell communications between microbes and their unwitting hosts, and then like a background noise all the critical day-to-day housekeeping duties that keep the bacteria happy and healthy and as part of a whole continual process. Additionally, because they are so small, fewer than 50 amino acids in length, it is highly likely that the proteins will fold into unique shapes and that these may well represent previously unidentified biological building blocks. If the shapes and functions of all these proteins can be recreated in a lab, they could help researchers advance scientific understanding of how the microbiome affects human health, and hopefully this could pave the way for new drug discoveries. A paper describing the research findings was published in August in Cell. Amy Blatt, Assistant Professor of Medicine and of Genetics, is the senior author. This was a previously known blind spot in observations, as the protein's minuscule size had made it virtually impossible to even identify, much less to actually study them. Well, using traditional methods at least. As Amy said, we've been more likely to make an error than to guess correctly when trying to predict which bacterial DNA sequences contain these very small genes. So until now, we've systematically ignored their existence. It's been a clear blind spot. Did you know? These little companions of ours account for far more cells in and on the human body than actual human cells do. They help with our digestion, supplement our diet, and generally keep us running at our peak, hopefully. But up until now, we never really knew much of who did what or said what to whom. So it was postulated that maybe answers might be found in the smaller proteins that have always been ignored by the old studies. And to top it all, these smaller proteins can slip through the cell membrane to ferry messages or threats to neighbouring host or bacterial cells, much more freely than the multitudes previously studied. So today we're in effect been eavesdropping on the internet with a paper cup and a string. So how did they solve this conundrum? See, in the past we would only identify the presence of protein coding genes by searching for known start and stop signals that we understood as denoting the beginnings and endpoints of a gene. And for larger proteins this served us well. But as the size of the gene decreases, the error rate rises and it starts becoming too contaminated with background noise to see the patterns accurately. Then a new approach to the same data sets changed all that. Vero decided to compare the potential small protein coding genes among many different microbes and samples. Those that were identified repeatedly in several species were more likely to be true positives. When this methodology was applied to ever larger data sets, they found not just the hundreds of genes they had all been expecting, but tens of thousands, orders of magnitude greater. To date, the proteins predicted to be encoded by the genes have been sorted into more than 4,000 related groups or families, and we can only guess where these new leads will take us. Obviously, they are now working with collaborators. Why does saying that word sound so dirty? Anyway, yeah, they're working with collaborators because there's so much to study, and it will be offering new insights for quite some time into the future, such as antibiotics or drugs for human use. In fact, as small proteins can be synthesized more rapidly, it could be that they are used by the bacteria as biological switches to toggle between functional states or to trigger specific reactions in other cells. Additionally, they are also easier to study and manipulate than larger proteins, which could facilitate quicker drug development timelines. So who knows what this could yield over the next decade? Anyway, time for my thoughts on the bigger picture. Yeah, it's a tough one. I mean, what does this not touch? But in truth, one of the biggest things facing us in the coming decades is antibiotic resistance. Antibiotic resistance heralds a return to medicine in the Dark Ages, where small cuts kill. Most surgery becomes a lottery, if you even bother with it. A child mortality rate that even your great-grandparents saw as consigned to history. And, well, essentially the end of all things fun. But this new research could provide us with a new arsenal at our disposal, a new way to protect ourselves from the exponential growth rates of the wrong bacteria in the wrong places. And maybe we can be more targeted with our application from the very beginning this time. Less of the broad spectrum carpet bombs. And maybe also we don't just use it to increase profits on meat farms. But who knows, when money speaks we often only step in once the cracks in the proverbial dam are beyond repair, with the wealthiest nicely relocated to the proverbial higher ground. But hey ho, what do I know? What do you think? Am I right? Am I wrong? Try to convince me with your well-reasoned argument, supported by your incredible evidence, and all from irrefutable sources. And I will see you next time. Thanks for watching.